Thank you, Professor Thalal, for the kind introduction. I also want to thank Claude Lecomte and Michael Zima for organizing this excellent legacy session and for the invitation to participate in this wonderful event marking the IYCR. It's exactly 1 o'clock in my watch, and I know I stand between all of you and a very sumptuous, delicious Moroccan local cuisine lunch. And so I'm going to be brief. We had excellent illustrations of very intricate, extended, high symmetry, and very, very beautiful patterns in the previous lectures, and also all around in this room. As you can see, there is no word symmetry in my title. I don't work on symmetry. I'm also not a crystallographer. So I'll try to do the best I can to hold you from your wonderful lunch which is waiting for you. Sorry. Yeah, so I work on crystal engineering and more specifically on pharmaceutical materials, and drug molecules actually are sort of an antithesis of symmetry. They are floppy molecules, large molecules, almost lacking any kind of symmetry except molecules such as hexamethylbenzene or cyclotriphosphazine, which organic chemists would consider as highly symmetric molecules. Most of the drug molecules and the examples that I will show lack any kind of symmetry. And so the elements of symmetry that you see in my talk are sort of retrofitted after I got the invitation from Claude, and he said this should somehow match up with this session, and I hope I can say something which falls within the theme of this session. This talk is about pharmaceutical and more specifically about solving solubility issues in pharmaceutical, which seems to be a current challenge in the drug industry because as the numbers state, over 80% of drug molecules pose a solubility issue. The good news is that in the last decade or so, it has become clear that crystal engineering perhaps can possibly provide solutions to some of these challenges through crystallography, through making solid forms which exhibit higher solubility and dissolution rate. And this has been covered in some of these. Now, the work of Amidon at Michigan has essentially led to this classification of drug compounds which have a hydrophobic part, which is the lipophilic part, which gives permeability to the drug, that is binding to the receptor, and the hydrophilic part or the hydrogen bonding part of the drug molecule, which gives solubility. And obviously one needs a balance of both in an ideal drug molecule. But unfortunately, as you see, those compounds are relatively small in number. The major contribution or the major categorization of drug compounds is those with low solubility and either high or low permeability. And within that, the class 2, which is amenable to crystal engineering solutions, that is the low solubility, high permeability is about 70%. So this talk will show a possible way to address some of these issues. And as I said, I will try to bring in symmetry to the extent I can. Some of these numbers kind of act as a guide. Less than 20 milligrams per liter is considered as low solubility. Between 20 to 60, 70 is medium, and anything more than 70, 75 milligram per liter would be, or microgram per ml would be considered as high solubility. Now, this table was about a decade ago. If you come to the present time, the trend is really changing. The percentage of drugs, and this is taken from a review of Abu Sirajuddin, who worked at Novartis almost his entire career, and he said that while a value of less than 20 microgram per liter for solubility of NCE, which is drug compounds, was practically unheard of till recent times. This has changed, and even 1 microgram per liter per ml or less is now quite commonplace. Now, drug discovery, of course, is a very, very long marathon, and the issue of solubility, ADME, tox, bioavailability essentially come up at the very, very end. Now, there are a variety of reasons why we are now facing drug molecules or seeing drug molecules with very low solubility. And I think I'll just share a few slides to show you what could be perhaps reasons. These are drug compounds which were approved by the FDA in 2011. And as you can see, some of them, particularly this one, actually lacks any hydrogen bonding groups. And that is the kind of functional group which would give water solubility. So these are not only rather complex, they have multiple functional groups, they have conformational flexibility, they certainly lack in any kind of symmetry. And many of them have very delicate functional groups, such as the one you see here, an ether functional group next to a trifluoromethyl group, which essentially means that making a salt with a very strong acid for this amine kind of functional group would be practically impossible, because the molecule would not withstand the harsh conditions of salt formation, which is a standard practice in the. They somehow also very often defy the golden Lipinski rule of 5, and while they exhibit very tight binding, almost nanomolar, their drug delivery is really a challenge. Now, this is 2011. Fast forward to 2014, which was a real bonus year for the pharmaceutical industry, 41 new drug approvals. This is sort of the Mount Everest of what I would call as a small molecule drug. Of course, biologic drugs can be even much bigger. This would be a small molecule synthetic drug approved in the last year. Clearly, this is far away from drug molecules structures, the types you would have seen in the 80s and 90s. So these molecules are the kinds which are, contributing to the low, very, very, very low solubility issues which is faced by the pharmaceutical industry. So this is where crystal engineering comes in, the area that I worked in, and the concept of the supramolecular synthon is well known to the audience in this room, advocated by Gautam Desiraju in the 90s. And there are essentially two kinds of synthons here, which are hydrogen bond motifs, which are associated with a given functional groups. This is the carboxylic acid, which makes the dimer, or we call it the homosynthon, between like functional groups. This is the carboxamide dimer, or the amide homosynthon, again between like functional groups. This would be a heterosynthon between a carboxylic acid and a pyridine, and that essentially means that you can make partnership between like functional groups as well as between complementary functional groups, such as acid pyridine or amide acid and so on. The numbers below derive from the Cambridge Structural Database, essentially give you an idea of what is the likelihood or probability of occurrence. And so if you have a high success probability, then that is something you should incorporate in your design element. Zavorotko is essentially the champion of this application of crystal engineering synthon concepts in pharmaceutical development and making improved medicines. The sulfonamide functional group, which is quite common in drugs, almost 50% drugs have the sulfonamide functional groups, somehow was missing from this library of the crystal engineers toolkit, and we have contributed in recent years, including an IUCRJ article which is in press on the heterosynthons of the sulfonamide group, which will allow one to engineer cocrystals and other adducts, related adducts, pharmaceutical adducts for sulfonamide kind of drugs. So this is just a brief outline of the kind of materials one sees in pharmaceuticals, crystalline materials. One can have polymorphs, which is different ways of arranging the molecule, and of course one can have the amorphous state where really there is no short range or long range order. This of course has long range order. Then one can have a second component just like the bow and the tie. One can have solvates, co-crystals, complexes, depending on whether the black dot or the second component is a salt former or a co-former or a solvent or a water molecule. One can also have solid solutions when there is a bit of shape similarity or isomorphous nature between the two components, where one can have a variety of stoichiometries. These are more like fixed stoichiometry, multi-component adducts. These are variable or continuous stoichiometry. The last of these is where the element of symmetry comes in the discussion that I will follow up, which is the eutectics, where essentially in a long-range sense, there is no change in the diffraction pattern or the periodicity or the long-range order, but in a short-range sense, there can be multiple phases of these two components, the red and the purple, where they can have an alpha phase or a beta phase or multiple phases thereof. So one can have polymorphs, one can have cocrystals, one can have solid solutions, and one can have eutectics. And now I will try to bring in symmetry in trying to connect these. So this is just the IUCR definition. Isomorphous crystals are essentially those that have same space group, unit cell dimensions, and one can do isomorphous displacement. And isomorphous crystals, of course, have a good likelihood of leading to solid solutions. Isostructural is something where the structures are similar, the space groups and detailed symmetry elements can be different, and so one can have isomorphous and isostructural. And how these sort of quasi-symmetry ideas could be applied in pharmaceutical materials is what I would like to share with you. I think Jenny already introduced this. There was a stamp which was launched by India, which has curcumin on the picture. And of course, the molecule needs no introduction. So I will just give this as a case study to walk you through some of the work that we are doing, and also how the ideas of crystal engineering and symmetry and isostructurality and isomorphous replacement can be used in making improved pharmaceuticals. So this molecule, of course, is well known. It has a large array of biological properties and medicinal activity. However, it is not approved as a drug because of very, very low solubility, less than 8 milligrams per liter. It has a very short half-life of less than half an hour and very, very poor bioavailability. So while it works very well when taken with your food, it's a great appetizer and a great food additive. By itself, as a pure compound, it really is challenging to deliver it in the body. And so our goal was to see if one can improve the bioavailability and solubility of this herbal ingredient. That's the molecule we saw earlier in the morning. So we started off with polymorphs. One crystal structure was already reported. We found 2 new polymorphs, and then we said, while polymorphs are good in terms of improving solubility and bioavailability, the main challenge with polymorphs is that the metastable polymorph, which has usually the higher solubility, can transform to the more stable one, and we found that within a matter of few hours, both form 2 and form 3 transformed to form 1. So really, in a practical sense, there was no advantage other than knowing that there is a polymorphic behavior for curcumin. So these are the powder patterns which we got for the bulk materials, which match very nicely. There was also an amorphous phase, and then the solid state NMR to further characterize the polymorphs. Knowing that, or observing that they have very limited stability in a practical sense, the 2 polymorphs, we then looked at cocrystals. So these are cocrystals, as I said, the bow and the tie, of curcumin with resorcinol and pyrrolidinol. Both of these are again bioactive molecules, and our idea was to combine the phenolic functional groups of the herbal drug with the phenolic functional groups of the coformers, which would perhaps not only give it higher solubility, but perhaps some synergy in terms of drug action as well. So our idea was, can we make stable soluble curcumin for an oral formulation via the cocrystal route? Again, these are just X-ray crystal structures to confirm that these are so this is the planar part is the curcumin part, and these are the connectors, the resorcinol here and the pyrogalol there, and just matching with the line pattern. So this is the solubility profile for form 2 of curcumin, which is faster dissolving than form 1 or the reference curcumin, and the amorphous one sits in the middle. This is somewhat surprising, but the main reason is the transformation of the amorphous to crystalline is perhaps faster than that of form 2 to crystalline, and this graph is for a short period of about 4 hours. After that, they all tend to convert. Now, this improvement or enhancement is about 4 times to the reference curcumin, whereas with pyrogalol and resorcinol, this line is somewhat light, but you can see the trace. It goes to about 40 on the y-axis. So, from an enhancement of about 4 for the polymorph of curcumin, we saw an enhancement about 40 times to the reference drug for curcumin and pyrogalol. Stability-wise, they are quite stable, both resorcinol and pyrogalol cocrystals of curcumin. And the enhancement is 40 times, which is good, but really not great enough to move forward in a long-term sense. And so the next idea was to look for other partners which would perhaps give us greater enhancement in solubility. And this is where we came across somewhat of a surprise, and that becomes the basis of today's discussion as well. So then our idea was to look at other co-formers such as nicotinamide, ferulic acid. These are again compatible biomolecules, hydroquinone, para-hydroxybenzoic acid. And as you can see, again we got solubility enhancement anywhere between 6 times to 10 times to 3 times. However, none of these co-formers gave us a co-crystal, that is a discrete adduct with a fixed stoichiometry and a different diffraction line pattern, that is a molecular packing arrangement from the starting components, which is curcumin and any one of the co-formers. What we did get were eutectics, which means they had a different melting point, a lower melting point, but the X-ray line pattern was matching peak to peak. And at the time we were doing this project about 4 years ago, DSC was perhaps the only and the most single method to characterize eutectics as opposed to co-crystals. Later on, we came across other methods such as pair distribution function analysis, and that is something that work is still pending. But what we did find out was that one set of co-formers, that is resorcinol and pyrogalol, are giving us co-crystals, whereas another set of co-formers, some of which are phenolic and very similar in structure, are actually giving us eutectics. So that was the next question we asked ourselves. What is it that is the transition point from getting a co-crystal to getting a eutectic? Because in terms of hydrogen bonding, in terms of molecular packing, they are quite different. The eutectics really don't have a long-range arrangement of molecules which is distinct from the original phase, whereas the eutectics have a difference at the short-range level, but really not at the long-range level. So this is where the idea of solid solution also comes in, and that's why I thought I'll bring in all those topics. The X-ray crystal structure of a co-crystal is different from that of the individual components, whereas the unit cell of solid solution is similar to one of the components. Eutectics, we believe, and that's the sort of philosophy we have advocated in this review article, eutectics are closer to the solid solution in that their crystal arrangement is similar to one of the components, but they are different in terms of their structural integrity because there is a second component. A solid solution possesses structural homogeneity throughout the structure. It is a single phase, but a eutectic is heterogeneous ensemble of individual components, and the differences are, as I said, only at the short range. So these are more like discontinuous solid solutions or phase separated material. Thus a eutectic may be better understood as a conglomerate of solid solutions, and this is really the sort of take home lesson from our study of a few cases, that if we combine two molecules, that is curcumin and a co-former or a bow and a tie, we get a co-crystal or a solid solution or a eutectic, and that result will depend on the nature and strength of the interactions between the heterosynthons or the unlike functional groups. If they are very strong and they are very specific and very directing, then one will get a co-crystal. And whether the two molecules have similar geometry or different geometry will kind of be overridden and dominated by the strength of the interactions. Whereas when the structures of the two components, the bow and the tie, are similar, and if the interactions are weak, then one will get a solid solution. And if the structures are different, geometrically they are very different from each other, like curcumin and resorcinol, or curcumin and nicotinamide, then one will get a eutectic. And I will sort of illustrate this through one more example. This is fluoxetine hydrochloride, which is a well-known drug, and co-crystals of this have been made with Fa stands for fumaric acid and Sa stands for succinic acid. Now, if you will see here, all the interaction in fluoxetine hydrochloride are of two types. One is an NHCl minus and the other is a CHCl minus. And that is, they are all sort of flanking around the chloride ion. On the downside is ethambutol hydrochloride, which is another drug, where all the interactions are of a single type. That is, they are either, all of them are of the strong type, either NHCl minus or OHCl minus. So fluoxetine hydrochloride is NHCl minus and CHCl minus, whereas ethambutol hydrochloride is either NHCl minus or OHCl minus, both of which are strong. When you add a coformer like succinic acid or fumaric acid, the CHCl minuses are replaced by the OHCl minus. So you are going from weak to strong, and we get a co-crystal. Not we. It was reported by Scott Childs and his group. They got a co-crystal. Ethambutol hydrochloride was our study. One strong hydrogen bond, that is NHCl, OHCl of the drug, is replaced by another strong hydrogen bond of the coformer, and we get a eutectic. So it's how much you are able to dominate through the supramolecular heterosynthon interactions that determines whether you get a co-crystal or an eutectic. So what about solid solutions? Solid solutions will happen when the two components have very, very similar shape and geometry. And an example is benzoic acid, fluorobenzoic acid. So these are isomorphous structures. If you mix the two, you get a solid solution of varying composition. If you take benzoic acid, pentafluorobenzoic acid, the interactions dominate because of the pentafluorobenzoic acid, and you get a co-crystal. If you take benzoic acid, benzamide, you have kind of lost out because you neither have, you have similar shape but no strong interactions to extend the lattice, and you just get a physical mixture. So the gradation is somewhat gradual, which is dictated A, by chemical interactions or the hydrogen bonds, B, by geometry or shape and overall size and symmetry of the two interacting partners. The same empirical model was elaborated with also compounds such as nicotinamide and isoniazid, where again we had this is not done in my lab, but by my student when he was, after he joined Professor Guru Rao's lab at IISc Bangalore as a postdoc, where he continued this work. So this is a gradation from succinic acid to succinamide and to the hemisuccinic acid succinamide. So it's a gradation from carboxylic acids to one acid, one amide, and to two amides. So the strength of the interactions is being varied gradually as you go from left to right. And this is the results with nicotinamide. The first one, the succinic acid, is always giving a co-crystal. The second one, in the case of nicotinamide, gives a co-crystal. And the third one, which is amide, gives a eutectic. In the second case, isoniazid, where the interactions are somewhat weaker, the first partner, which is succinic acid, gave a co-crystal. Second and third were eutectics. If you go even weaker by looking at bipyridine as a partner, where the interactions become even weaker because it lacks a strong hydrogen bonding functional group, the first combination with succinic acid gave a co-crystal, whereas the second and third became physical mixture because the interactions were not sufficiently strong to hold the structures together. So while the model is empirical, it does tend to teach you some lessons as to how to make combinations and why one would expect a co-crystal in one case and a eutectic or a physical mix in the other. Of course, as I said, a thorough characterization of these eutectics is still pending in our lab, and the way to go perhaps would be to go to the high energy synchrotron sources. We heard about the developments and the future in this area yesterday evening lecture. So the idea would be to not look at the intensity versus the 2 theta plot, but to look at the radial distribution functions. And this is where at the short range of 8 to 10 angstroms is where one can see some of the differences between. This is a carbamazepine example between the nanocrystalline material of carbamazepine and the 3 polymorphs. The difference is indistinguishable if you look at the 2 theta plots, but if you look at the radial distribution plots from the pair distribution function analysis, then one can clearly correlate that the nanomaterial is matching with CBZ form 3. So this is the kind of analysis which we still need to do. It's more of a reminder that we have to take up our eutectics and do a similar PDF analysis. Sorry. Just to sort of update you on the curcumin story, we did follow up after the eutectics by making a combination of curcumin with artemisinin, and here we got something because the molecules are much bigger and much more floppier. We neither got a cocrystal nor a eutectic. We got a coamorphous solid dispersion, because the only kind of interaction that one can visualize here is a single hydrogen bond from curcumin to the carbonyl group of artemisinin, and this is the coamorphous material whose FESEM image is shown to the bottom right. This is curcumin, this is artemisinin, this is the physical mixture. So it's a highly nano-sized particle distribution in the coamorphous curcumin-artemisinin 1:1 mixture. And this is the bioavailability graph. I know this doesn't fall into the main theme of this session, but I just thought I will show you that we did kind of reach the target which we were hoping to get, which is a high bioavailability and dissolution for curcumin. This is 1200 nanogram per ml, which is about 1.2 microgram per ml, which is quite a big improvement compared to curcumin, pure curcumin, which would almost fall at the baseline on a curve like this. The important thing is that this bioavailability study is done in rats, and the important thing is that the Cmax occurs at T max of 30 minutes, which ties up very nicely with the half-life of curcumin, as I showed you right in the beginning. Curcumin has a half-life of between 30 to 45 minutes to 1 hour. So you would want maximum exposure in less than 1 hour, and this is really what seems to have been achieved with the curcumin-artemisinin. And the last example is from Desiraju's lab, where they use the idea of structural mimicry and isomorphous displacement. This is again orsinol. I think we saw listed as one of the articles in Samar Hasnan's presentation from his lab in collaboration with Guru Rao in IUCRJ. This is another paper on the same topic, where this is a binary co-crystal between orsinol and bipyridine, and then there is a second bipyridine which is sort of floating free. So it's a 2:3 combo, of which the two are tied up here and the third one is clearly distinct in terms of its bonding. And so they replace this free BP with a second component which kind of matches in size and geometry, and a large number of coformers were very freely and smoothly replaced with the free BP to go from a binary co-crystal to a ternary co-crystal, which means one can have, by design, 3 distinct chemical elements integrated into the same crystal lattice. This is phenazine, phenanthrene, anthracene, and thiophene, biphenyl, and so on. All these are sort of isomorphous and isostructural to bipyridine in that they are matching in size and shape, and in a repetitive way, the free BP was very nicely replaced to give this family of 13 or so ternary co-crystals, which are all isostructural and isomorphous. I will close by extending the invitation extended by Gautam Desiraju in his video presentation. I would like to invite you to the next IUCR Congress, which will be in Hyderabad in 2017. We hope to see all of you in this room, plus many, many times more your friends, colleagues, collaborators, and all of you here. Last but not the least, shukram to the Moroccan hosts.